It's my pleasure to introduce Tanya White. She and I have collaborated for many years, I guess, a lot since I was in New Mexico. She did her medical degree from University of Illinois and her PhD at Erasmus University in Rotterdam. And she spent a number of more recent years. She was at University of Minnesota, then went back to Rotterdam, and she was kind of heading up the imaging component of the Generation R study, I believe. And now she's at the National Institutes of Mental Health, as you can see in the section on social and cognitive developmental neuroscience. And she does, pediatric studies of pediatrics. She does neuroimaging and population neuroscience. And I'm really looking forward to hearing what you have to share with us today, Tanya. So I'll let you go ahead and take it from here.
Well,
thanks for joining. Yeah.
Thanks for the opportunity to share some of the work. And I did move a year and a half ago to the NIMH. And I'm kind of putting together studies here. But what I'm going to be presenting today is not data that I've collected yet at the NIMH, but goes into the 13 years that I spent at Erasmus University Medical Center in Rotterdam in the Netherlands. And it is I'm going to be talking, giving an overview of pediatric population neuroimaging, and then I'm going to take a detour, not really a detour. I'm going to describe how I've been thinking about stochastic processes in brain development and how that could have a potential public health benefit. And that'll come near the end, and it'll be a story associated with the effects of folate during pregnancy, low folate, and subtle differences in brain development. So what is population? And my focus has been pediatric population neuroimaging. It's at the intersection between epidemiology and neuroscience, and in my case, developmental neuroscience. It's this area here where there's the intersection, which is really focusing on aspects which can affect the population as a whole. And genetics is also a part of this, especially when you consider a number of psychiatric disorders. I'll be talking a little bit about autism, which has heritability rates between 0.6 and 0.8. But it's this area that I'm going to be describing, this intersection between epidemiology and neuroscience. So imaging in epidemiology, on one sense, when I look at some of the old work in epidemiology, it actually goes back to the beginning. So let me show you the very first imaging study in epidemiology in the mid-1800s. And this is John Snow. And these blue dots here are wells in the Soho district of London. This is he's one of the founding fathers of epidemiology. And the red dots gives a number of individuals who died due to a cholera outbreak. And so what John Snow did was kind of on a map look at the deaths and find out that this well was actually the one that was infected. There was like a dirty diaper that was leaking cholera. And so most of the deaths were surrounding this well. What I've not done is to quantitate how, you could do a quantitative image, and he didn't do that. He just looked visually. But this is a 2D imaging study from many years ago. So let's move on to what I'm going to be talking about quite a bit. I'm going to give you an overview of what a population-based study is, but really how the 9 months is a period of time that's critical in development. And there's studies called the fetal origins of health-related issues, including mental health. So let's go back in a review of what happens in, first of all, early development. So before 2 weeks, you have a single cell that continues to grow. You have the ectoderm, the endoderm, and the mesoderm. The skin and the brain both come from the ectoderm. And roughly at 2 weeks, you've already had the neural plate that folds, that becomes the neural tube. And there is symmetric subdivision around the ventricular zone. So here's the neural tube, it's folded. Here are the neuronal precursors, and they undergo symmetric subdivision between 2 and 6 weeks. And at 6 weeks, it changes to asymmetric subdivision. But I want you to think about this period. It must be under very tight genetic control, because I remember what my father said when I was little, when I was 5 or 6 or 7, around that age. He said, so let me give you your allowance, and I can give you either a penny a day and double it for this month, or I'll give you $1, $1.50, whatever my allowance was. And of course, at a certain point in time, I did the calculations and realized that if I did the doubling every day, that in a 31-day month, I'd be a multimillionaire. So this is a similar principle with symmetric subdivision. For every cell division, you're doubling essentially the number of neurons that are going to populate the cortex. At 6 weeks, there's a change, and then it moves into asymmetric subdivision, where one cell becomes a neuron. It differentiates into a neuron. The other one remains a daughter cell and continues to divide to populate more neurons. These neurons along the ventricular zone, they migrate in an inside-out pattern along these radioglial guides, forming the cortical thickness. So if the brain in animal studies, and believe it was done in sheep, where they irradiate the brain during this period of time, you'll have decreased cortical thickness, but not decrease in the volume. So it's a specific window where the thickness of the cortical surface is determined through this period. Then as you move on to the 3rd trimester, the brain changes from a smooth lissencephalic or structure or morphology, and most of the gyrification occurs between 24 weeks and birth. And at birth, even though the brain is about 1/3 as small, it looks very much like you see in adult brain. And actually, if I show the entire pattern of gyrification of the human brain, you can see that it goes from this smooth surface at 5 months. You start seeing some of the deeper fissures starting to emerge, and then by 9 months, you have primary central sulcus, Sylvian fissure, and all the secondary fissures, as well as some of the tertiary fissures, but they grow fairly dramatically as well, in early life. In fact, the deeper sulci tend to be more genetically driven compared to the tertiary sulci that are influenced quite a bit more from environmental factors. Okay, and so I've talked about what happens up to birth here. And this graph here is, for those of you who kind of study development, it's not uncommon that here's 1 year of age, and this graph that shows brain aspects of brain development, 1 year takes up half of this figure. But you see other, so myelination, and it doesn't just suddenly jump up at one point, but there are, it starts essentially during the third trimester and continues on beyond adolescence and into early adulthood, maybe even into the 40s. And then you have development of the cortices that occur initially with the primary visual, auditory cortices. Then you have the secondary cortices, perception, language, and then finally the last to mature is the higher order cognitive functions, association pathways, pathways connecting the different lobes and the prefrontal cortex. And this says the brain is kind of done at age 15, but that's not true. We know it continues developing much longer than that. Similar to this slide is a slide looking at white matter development. This is a classic 1967 paper from postmortem study by Yakovlev and Lacours that show that the development during fetal first year of life, and essentially at 4 years of age, most of the white matter pathways have been laid. They're still remodeling, but by 4 years, most pathways have been myelinated, except you have the reticular formation, which you might think of attention, and then the association cortices, which goes on. And in this 1967, third decade of life, there's even a question mark here. Is this continue on? And some of the fMRI or the DTI studies have shown continued white matter or increases in FA into the fourth decade of life. So I will be talking in a bit about stochastic processes, but I want to think about this think about building a brain from an engineering perspective. And you're given about 7,000 to 10,000 genes that that code for the essentially 80 billion and 100 trillion neurons that make up the brain and all the connections, and not even, considering some of the other elements, the glia, the microglia, the immune mechanisms that occur. So this is quite an interesting problem, and the way the brain captures this is through redundancy. So I mentioned symmetric subdivision. It must be under very tight genetic control because one additional cell division for all the cells, and you've got twice the number of neurons in the brain. But within these pathways of cell division, there are times when you get a mismatch just by random Brownian motion, a mismatch between certain molecules. So for the smaller molecules, it's not a problem because there's a mass action effect and it washes out. But for some RNA, there's only 10 of a certain type per cell. And so then the probability that small deviations early on can have an effect. Similar to the neuronal migration, there's different pathways that could be taken, and that those pathways could also affect the connectivity between different regions. So there are, even though there are boundaries that are placed for what is allowed, there is some room, and there must be some level of probability that takes place. So there have been a number of people who have talked about articles on stochasticity, a part of what I'm going to show you here. So this was George Davey-Smith, who's heads up the ALSPAC study in Bristol area of the UK. And he talks about this gloomy prospect of embracing randomness in population health research. The good news is, by the end of my talk, this gloomy prospect, I don't think it's all that gloomy, but we can talk about that. Peter Clark wrote a paper, The Limits of Brain Determinacy. There was this interesting paper in 2020 where they looked at, in fruit flies, they looked at the visual system, the dorsal cluster neurons, and found that they were, tried to inbreed them, but they couldn't. And this mechanism was completely stochastic in its development. They then intervened and could alter it, which was very unique and interesting study. And then this paper, which was really, Klaus Gartner wrote about his 30-year-long effort to standardize laboratory animals. The thought was, if you can standardize laboratory animals by having them have the exact same genetics and the exact same environmental, control the environment to be identical, then you could control something like the liver or the spleen or the lungs or the brain or something so that it would be identical in size. And then you could very carefully titrate environmental changes to see what effect they would have. So this was his career, and he essentially said, I couldn't do it. No matter how much I tried to control the environment and genetics to keep everything the same, there's this random variability. And in fact, there was very little difference whether the mice were raised in a very controlled environmental setting or if they were raised wild type where they had to forage for their own food, be afraid of being eaten by owls and other things where they put them in a different environment. So this is one of the things that I think is it's tremendous for inducing diversity in the population, but it's challenging from the standpoint of precision medicine. And then finally, Günter Vogt also wrote some papers about stochastic development variation. And if anyone has questions or wants me to share some of these papers, let me know. So what I'm going to do now is I'm coming back to stochasticity in the future because that's a story that emerged with folate. But I'm going to give an overview of a population-based study, a pediatric population birth cohort in which actually they were recruited during prenatal life. And some of the results that we've seen from pediatric population neuroimaging. So the Generation R study is a prospective cohort. So between 2002 and 2006, about 10,000 mothers and their children, or the mothers were pregnant or had just given birth in a small percentage. And the goal was to study these children, and they're now into young adulthood right now. What makes it epidemiologic is that the inclusion criteria was to be within this blue area. Here's Rotterdam. To be within this area, and the mothers were pregnant. About 60% who were approached agreed to participate. So Rotterdam is a large port city, and it's quite ethnically diverse. about 58% Dutch and other Europeans. So we have a lot of families from other racial and ethnic backgrounds. Now I'm going to walk you through really briefly just some of the measures that we collected in the study, and the ones that I'm going to be talking about later. Our first measures of the brain was ultrasound measures. So in the first, second, and third trimester, we had collected or orbital frontal cortex, OFC, orbital frontal circumference, biparietal diameter, and then the second and third trimester, we could also collect cerebellar, a distance of the cerebellum. And then after birth, in a subset of the individuals, there through the anterior fontanelle, you can measure different characteristics, ultrasound measures. For example, this blob here is called the gangliothalamic ovoid, which can be measured. It gives a measure of subcortical brain development. And you can look at the ventricles and the corpus callosum as well. Blood has been collected at many times during the course of the study. Questionnaires, I often say that the parents should have an award for the number of questionnaires that they filled in over time, because it began during the prenatal period and perinatal with demographics, lifestyle, looking at psychosocial characteristics, and then postnatal development as well, from behavior, sleeping, activities such as sports and watching television, cognitive development, and psychosocial characteristics. One of the primary behavioral measures that was systematically collected from beginning at 18 months, 3 years, 6 years, 10 years, 15, and 18 to 20 was the child behavior checklist. So that's something which was longitudinally collected. And there's been measures of cognitive performance, including a nonverbal IQ in all the children when they were between 5 and 7. And the 13 to 15 wave had the WISC-5 done on the entire cohort. For the first imaging wave, we did NEPC-2, which is a neuropsychological assessment, and that was in a subset of individuals. And there were also interviews performed during the perinatal period over depression, and also the Berkeley puppet interview. And I'll just show you briefly a brief description of what the Berkeley puppet interview is. And that gives me an opportunity to have some water.
Hey, hoe heet jij? Hoi Leliana. Hey Leliana, hoe gaat het? Ja, met mij ook. Mij ook. En ik heb niet veel Engels. Ik wel veel Engels. Niet veel. En ik heb veel vriendjes en vriendinnetjes op school. En ik heb niet veel vriendjes en vriendinnetjes op school. En jij, Merel? Ja. Hier, als kinderen spelletjes spelen, en dan mag ik niet meedoen. En als kinderen spelletjes spelen, dan mag ik wel meedoen. En weet je zelf, als ik op school ben, dan mis ik mijn mama of papa. Als ik op school ben, dan mis ik mijn mama of papa niet. En jij? Ik kan niet, omdat ze komen. Ja, precies. Dat is net als ik. Oh, niet veel. Ik hou wel veel. En jij? Ik heb ook niet echt veel. En soms dan sla ik mijn juf of andere grote mensen. En ik sla mijn juf of andere grote mensen niet. En ik bepaal niet. Weet je, ik vond het wel heel gezellig om met jou te kletsen. En we gaan even kletsen hoor. Tot veel plezier hè vandaag. Hey, dag Demi.
So what I like about that is it gives an idea. That was that Berkeley puppet interview was done in about over 5,000 children, and you can see a bit of a variation, and it gives you an idea of what the population-based study is. It's a section of within the hospital where there's multiple rooms, and the children come in and they go from room to room. And it's not just mental health. There's pediatric pulmonology, pediatric dental, cardiology, ophthalmology, growth and development. And my role was in this large study was the brain. And so I'll walk through that briefly. The fourth wave, I left a year and a half ago, and Ryan Metzel has taken over my position there, and they're continuing on to scan. So we started in September 2009. Our first wave was completed in July 2013. We had 1,070 6 to 8-year-old children. That was not completely population-based. For the first study, we used a hospital scanner, and then in 2013 we had a dedicated scanner. I'll show you a little bit of the difference. So the first wave, we had specific grants to ask specific questions, and by 2013, with the arrival of a dedicated scanner, that meant we could invite the entire cohort. And for that, we scanned over 4,000 children. And at one time, we were averaging 130 to 100, during the course of the imaging, 130 to 170 children per month. Wave 3 was in October 2016 to 2019, and that was almost 4,000 13 to 15-year-old children. There were some challenges during that scanning session due to braces. It's one of the challenges when you have good health care good dental care for children under 18. And so about a third of them had braces. And then wave 4 began in 2021. COVID resulted in some delays, as it did for, I think, many of you. So we had 30 minutes to do our scanning, so we had to choose wisely. We did a T1 weighted structural image, 35 direction DTI, and 6-minute, 2-second resting state scan. And the other thing is that we worked hard to try and keep the scanner as stable as possible. You have the physicists who want the latest and greatest upgrade on, and so, but since it was a dedicated scanner for research for a longitudinal study, the Generation R study, we only did safety upgrades during the course of the study. When you're scanning children as young as 6, there's challenges with movement. So we had a mock scanner set up, and we had some toys that children could play with. To get used to the scanning environment. And then one of the PhD students actually made this 2 different graphs and showed the children. They said, this is how the pictures look of your brain when you move a lot. This is what they look like if you stay still. And we think that actually helped. We didn't do a controlled study to see if it did, but the thought was that they could visualize what movement did to the brain, and it seemed to help a little bit. So the other thing that we did was that we had a rating scale since we were doing young children, and this is one of the things the IRB wanted to make sure that, you know, that we had some level of consent, assent for the children, even though that they were, our first wave was 6 to 8. So we asked them how did you feel during the scanner, and were you anxious during the scanner? And if at any time they pointed to a 5, we'd stop. So they knew that, their parents knew that, and there were times the children pointed to 5 and the parents said, no, we want you to go on. But if the child indicated a 5, then we stopped the process. But children actually did really well in the scanner. So we did this rating scale before the mock scanner, after the mock scanner, and after the real MRI. And you can see that most children, a few children stopped. I guess the blue and the green, the purplish blue here, they were already done, so we couldn't stop. But what's interesting here, so here's the level of anxiety. So you can see the anxiety of the children decreased over time. But when you ask the parent and the researcher their ratings of the child, they're higher than how the child rated themselves. So we as adults tend to rate the children as being more anxious than what they actually rated themselves. So I'm going to move on. So there are, and I know I don't have all of them listed here, but there are some very large pediatric neuroimaging cohorts available. So this is population-based. So you have studies of typical development, a number of different studies, and what these studies, it's based on the inclusion criteria and the recruitment strategies. They tend to exclude children with psychopathology or family history of psychopathology in some cases. The clinical and high-risk studies are looking at individuals who already have a diagnosis or who are at high risk, either behavioral or genetic. You have a number of twin studies, but population-based studies really focus on how trying to catch a population that's representative of a certain group. And Generation R and Gusto in Singapore are two studies that really focused on beginning during prenatal life. There are other studies now that are focusing on that. And they tend to be oversampled for certain populations, such as opioid or drug exposure during pregnancy. So here's the Generation R. So what makes it a population-based study? Here's the total sample of recruited, and I have two different demographic features here. Dutch, non-Dutch, non-Western, and then non-Dutch Western. And maternal education. So here's our total sample. So I said wave 1, we actually had grant funding to look at specific populations. So it's a bit oversampled. It's less representative of the entire sample, and the entire sample was really quite representative of Rotterdam. So you can see some differences here in both ethnicity and maternal education. But once you get to wave 2, where our sample was much larger, it's a bit more representative of the general population. Not completely, there are some differences, but the goal is really to try and get your recruitment to match as much as possible to be representative of the population. One of the things that I guess in population-based studies and when you're looking at epidemiological approaches on a large scale, so you have different, this is A, B, and C. This is an illustration of the hippocampus in these three different groups. The hippocampus is the same size in A, B, and C, and it's much bigger in D. And this is just to illustrate global versus regional differences. So if you have a smaller brain but the same size hippocampus, or a larger brain and the same size hippocampus, and you're not controlling for something like total brain volume or intracranial volume, these will all look the same, and yet the brain has certain global characteristics. So generally, if the brain is bigger, the hippocampus is bigger, there's some growth mechanisms that involve the brain overall. And so generally, for smaller structures, I control for intracranial volume. So let me move on to some of the studies. I've kind of explained, given a teaser about stochastic processes, which I'll come back to. I've given a little background about population neuroimaging and what makes studies of population. Now, when I was in Rotterdam, I had kind of 5 major research lines. And I'm going to be talking about some of the prenatal exposures and downstream effects in offspring. That's really going to be my focus for this talk. And then focusing at the end really on the folate story. The nice thing about this first one is that there's a direct translation with the findings and public health, and you'll see that. But these large population-based studies can have other advantages. For example, if the sample's large enough, you can look at psychopathology across the continuum. For example, with autism, you can look at clinical, subclinical, or small amount of symptoms, and you can ask the question, is there a continuous relationship between, for example, some neurobiological feature, could be amygdala size, and autism and autistic symptoms? Or is it more of a dose effect that autism spectrum disorders there's no differences in the brain, it's non-linear up until you get a certain point in time, and at that point you start seeing changes in the brain. So that's psychopathology across the continuum. Emerging psychopathology is something with both high-risk studies and population-based studies where you can say, okay, what did the brain look like before someone actually developed a disorder? You can see how it emerges over time. This would be different than sometimes I think when individuals already have the disorder and you're trying to look back and put the pieces of the puzzle together. It's a bit like archaeology. You're trying to make sense of what happened, but it's always better to watch as it's emerging and to see what occurs when. Imaging genetics is something that we were involved in Rotterdam, which I won't be talking much here. And then, of course, typical development, because understanding how the brain typically develops is really crucial to make sense of what happens when there's differences or trajectories that are different. So let me talk about smoking during pregnancy. And smoking during pregnancy is, to what effect does that have on a developing infant? And we already know, the children, actually I think it was in the 40s and 50s, physicians told mothers to smoke so that their infants would have easier delivery. But fortunately, we have moved beyond that. So the first study that, actually this was before I came, was looking at prenatal ultrasound measures using head circumference and biparietal diameter and length of the ventricle, of the lateral ventricular volume or distance. And so there's 3 different groups here. This is gestational duration in weeks, up to 40 weeks. The dashed lines are those who continued smoking, and this is difference in fetal head circumference. The reference is the non-smoking, and what you can see is that those mothers who continued smoking during pregnancy had a gradual decrease in or greater difference in head circumference, whereas those who quit smoking when they found out they were pregnant had no difference. So this is one study, really, that has I guess it should be to some extent a common sense, but it's nice to show that if you're smoking and you become pregnant, quit, and you won't, the growth differences during pregnancy will not take place. So what happens downstream? So this was one of our first studies. We used 113 children exposed to prenatal tobacco and matched them 1-to-1 on age and gender. And we were using Free Surfer QDEC, looking at cortical thickness measures and volume measures on a surface-based approach. And we found fairly, I tend to call this more global differences, because if you decrease the threshold of significance, it actually, it's pretty much all in the same direction, affecting multiple areas or more global areas of cortical thickness of the brain. And this pattern also was very similar for those mothers who quit smoking when they found out they were pregnant. There was no difference, whereas the difference was seen in those who continued smoking during pregnancy. Now I'm going to walk you through some of the studies looking at autism spectrum disorders, and these are epidemiological studies. And I'll start with folate. We didn't find that folate levels were associated with autistic traits, but folate intake was associated with less autistic traits. So there's some evidence that our folate levels were measured late first end of the second trimester, but maybe it's those mothers who take folate before they're pregnant have greater resilience against autism, or it could be residual confounding, in that those who do take folate medications before they're pregnant might somehow be different than those who don't. Something always important to consider in these studies. There's been at least 6 studies looking at air pollution and the relationship between air pollution and autism in the US, showing differences, showing that air pollution does have an effect on the development of autism. 4 studies in the EU combined did not find any difference. Whether there's differences in the characteristics of pollution between the US and the EU is not clear, but we didn't find a relationship. C-reactive protein, which is a marker for maternal immune activation, there was an initial very strong effect, which was attenuated when you control for demographic and maternal health related factors, including BMI, although BMI is something that results in some mild immune, kind of low grade hyperactive immune response, and so that could also be a little bit of overcorrection. We found an effect between selective serotonin reuptake inhibitors used during pregnancy and the later development of autistic symptoms. And this group also included one in which mothers who were exposed to depression but not to SSRIs did not have the same rates. And then we've had 2 studies looking at the role of vitamin D between prenatal vitamin D levels and downstream autistic symptoms. So there's a number of different environmental factors that could be related to autism. But I was wondering about what if you think of environmental factors like you do genetic factors? So it's not one specific gene, but it's polygenetic that's associated with some psychopathology. And so what if it's polyenvironmental? So it's multiple environmental genes that each have a small effect that contribute to, in this case, autism. So funded by the Simons Foundation, I did an environmental-wide association study of autism spectrum disorder. And the rationale was a data-driven approach can yield new environmental factors associated with ASD. Environmental risk factors, as I'll show you in a minute, they cluster together. They're much different than genetic factors, which tend not, once you're far enough away from the base pairs, it's essentially random, but environmental factors cluster. Then what I mentioned before, to create a polyenvironmental risk score to test whether multiple environmental factors play a role. And then if you simultaneously test multiple variables, then you show both the positive and negatives, and you can reduce publication bias. So there's quite a number of advantages of this approach. So I would do this differently. If you have questions about this, I can talk about it later, but put over 1,000 variables, including questionnaire variables. If I were to do it again, I would start with just the biomarkers because it's much easier. But and questionnaires, you have potential for bias, shared rater variance, for example, how someone rates themselves may have certain characteristics of how they rate their children. But in any case, so this is a Manhattan plot of environmental factors, but these are not covariate. So these are univariate linear regression analyses. So, and the purple line is the Bonferroni. You can see that when you, so this is a huge amount of bias in this study, but we looked at 83 studies of articles in the literature, and what are the key covariates used in these studies for studies of autism or autistic symptoms. And you can see the relationship between these variables and the SRS, that is what we use for autistic symptoms, the social responsiveness scale. So you can see that most of them are hugely significantly related to the SRS. So sex assigned at birth is less than 10 to the minus 15th, so highly significant. This gives a demonstration of how correlated environmental factors are compared to genetic factors. These are a number of the covariates that we used, and you can see that they're related to many, many different variables. So this is one of the challenges in doing an environmental-wide association study, and probably it's best to consider clusters of data. But when you include all these covariates, the 8 different covariates, you can still see that there's a number of different markers that surpass Bonferroni correction, and whether those are real, whether it's shared bias variance, rater variance from how parents rate, or some other factor. So when you're looking at some of the questionnaire data, it's hard to parse out. It's not really environmental. Some of it can be genetics. For example, here are the top variables. And you see obsessive compulsive symptoms is the highest rated. That's questionnaire. And then other health, difficulty in decision-making, energetic, a global severity index. So most of the ones that were highly rated were questionnaires related to psychopathology. So in our biomarkers, we had some maternal fatty acids and a placental growth factor. Jolene Steenweg de Graaf wrote up the fatty acid relationships. So let me talk about a hypothesis. If you had on the x-axis, you have a poly-environmental risk score, and on the y-axis, you have a polygenic risk score, then my hypothesis, how I would think about it, is the higher you go on either of these, the greater the risk of having autism. So how does our data look within this window? And it's actually red dots here are children with a diagnosis of autism. Blue are the typically developing. So you see it's red in a sea of blue, distributed overall. But if you have a bit of an imagination, or not necessarily an imagination, because I'm going to help you out here, you can see that at least in this case, if you're below a certain polygenic risk score, and if you're below a certain poly-environmental risk score, then actually there were no red dots. So maybe this could be helpful to exclude certain individuals in the future. But other than that, the red is nested within the blue. Now, finally, I'm going to finish up and get into the stochastic. I hope I have enough time in my. It's about 5 minutes more, should be fine, and then time for questions. This is a really interesting question. What about nutrition during pregnancy? Can it promote optimal brain development? And then this question of prevention, can it prevent psychiatric disorders? So I'm not sure about the second, even though I'm going to show you a couple events that took place where there's some support for that. And I'll start with the Dutch hunger winter of 1944 to 1945. So D-Day took place in June of 44, and the Allies crossed the English Channel, landed on Normandy, and many casualties, and started moving north up through France, up through Belgium, and they reached a set of rivers and which had bridges that were challenging. In fact, there's a movie, main actor of that movie, one of them just died, can't remember his name, but A Bridge Too Far describes how they were blocked by the set of these rivers in the Netherlands. So the river, the Rhine, cut through, and essentially, and its tributaries cut off Rotterdam and Amsterdam and that area in which there was very little food during that period of time. And there was a very harsh winter, the winter of 44 and 45. So they were eating mainly bread and potatoes, less than 500 calories a day. There was double the rate of neural tube defects, and also a doubling in the rate of schizophrenia. So it's not necessarily nutrition, could be stress, other factors. I mean, malnutrition can have a huge effect, but there was something that occurred that doubled the rate of schizophrenia in the population who were pregnant during the Dutch hunger winter. There's other evidence from a famine that occurred in China with about 30 to 40 million deaths in this region of Anhui, if I pronounce it right, region of China, where again there was a twofold increase in the risk of schizophrenia and due to the famine that was taking place there. So I was curious to look into folate and ask the question. We know that low folate during pregnancy is related to neural tube defects, but what about the question is, even in the absence of neural tube defects, is it possible that low folate has subtle differences in brain development during prenatal and early life? So why folate? Because we can't synthesize it. I mentioned it's associated with neural tube defects, but it's involved in many, many, many different biological processes. In fact, if you look at the biochemistry of folate, the Krebs cycle is here, the energy, and it's connected to all of that. So it's involved not only in metabolism, but also repair of the genome, gene expression, myelin formation, neurotransmitter synthesis. So this is it's something that we can't get from, we can't synthesize. We can only get it from our diet, and it's critical. So the other thing is that due to laws in the Netherlands, where it was where you were not allowed to add any products to food up until the time of the EU, that changed when the EU came in. The Netherlands tended to be, in the period of time of the Generation R, the lowest in average intake of folate. And half the Dutch women didn't take folic acid supplements during pregnancy. So there was a deficit in folate. So our first study was related to prenatal brain growth and low folate. So 5,123 who had normal folate levels, 822 who had low folate levels. And the folate levels were collected during, at the time, around the end of the first, early second trimester. So we found that there was 0.1 millimeter less growth per week compared to the controls for those with low folate during pregnancy. So some people might ask, well, 0.1 millimeter less per week compared to controls. But if you do a little math and you say, okay, how's that translate into neurons? So you have 28,000 to 49,000 neurons per millimeter cubed, about 1,000 to 10,000 synapses per neuron, and it's about 1.9 million neurons and 1.9 billion synapses less per week, calculating around the mean third trimester circumference. So what happens later, 6 to 8 years later? So when I started this project, and if you asked me what my hypothesis was, I said, it's kind of a win-win situation. I think it's going to be gone, and I think it's a good message. We're going to say there's so much brain plasticity that occurs after birth that the effects that you see during prenatal life will be gone. So we had 256 Dutch mothers, 62 with low folate, using high-resolution T1, and we found those who had low folate had low folate during pregnancy or low growth. Those same children continued had low total brain volume 6 to 8 years later. We did follow that up with our third wave, and our second wave, where I mentioned we had many more participants, and essentially. Here's the difference at birth, around 8 years of age, and then 11 years of age. So this difference that's there persisted. And when you look at, okay, so we used 7 or 8 nanomolars per liter. But here's what you see when you choose different thresholds for cutoffs. Here's total brain volume. It starts to make its way back up, and also for cerebral white matter volume. So it was still there 6 to 8 and 9 to 11 years later. So this was a bit perplexing to me, because if you can have studies where they show juggling results in increased growth of gray matter, why doesn't it disappear? Why doesn't it catch up? It seemed to me that it should. So one possibility was those who have low folate during pregnancy continued to have diets that had low folate. So then I said, okay, are there other examples in the literature of studies where there isn't that catch-up growth that you would have expected? And there's a couple. So I'm going to focus on this, the one on the left, but the right is very similar. On the left is from the Bucharest High Risk Project. So this is a study that occurred in Bucharest, in which they had orphanages in which there was very little, it was a deprived environment. They had warmth, shelter, and they had food to eat, but they didn't have any type of social interaction. And so in Romania, they went into and randomized, this was a randomized control trial, to be either randomized to care as usual or to a foster care group or a normal control group here, typically developing Romanian children. So what I can tell you, the result from those who were randomized to the foster care group, especially before 2 years of age, had significant improvements in social and cognitive development, really striking improvement. And yet, when you look at the total gray matter volume between these groups, I'll say if you read the paper, it focuses on, there's a bias towards positive studies, but I think the negative study here, the negative finding of not showing differences, is more interesting. If these children in the foster care group had significant improvement, why then do they not show changes in something such global total gray matter? They had such global improvement. So here's what I'm thinking. We can't create more neurons in the cerebral cortex once we exit infancy. So I'm going to come back to this first 9 months. So it's best to assure optimum growth and development of the brain during prenatal and early life. And this is actually, right now, the best approach is by healthy eating, prenatal vitamins, limiting, not limiting all stress, but certainly large amounts of stress are not good. Now, there's not enough genetic information to code for the precise connections of all brain cells, so it relies on overproduction and pruning. And with the redundancy, it usually goes right. It usually works out. But one possible is if there's fewer neurons and synapses in early life, there's a slight risk that some of those connections that are really key could be pruned. And this is just a population-wide hypothesis, and I wrote it, kind of described this and a lot more background information in this paper, including having fun reading about the debate between Albert Einstein and Niels Bohr when Einstein said God doesn't play dice with the universe. Yeah, this was a fun paper to write, and that's the last part. You can't lose it if you've never had it. Is really, there's an element of chance, because we can't lose a neuron if you've never had it, but it's better to have it and lose it, being heavy in mechanisms, than never to have had it at all. Okay, the merging fields of epidemiology and pediatric neuroimaging. In what I showed you, I gave you examples where there's a direct translation into public health. And then I hope I gave an argument that prenatal life is definitely a critical period for brain development, because that's when a lot of the action takes place. And if we want to think about prevention on a population level, on an individual level, it's a different story, but we might want to think of that period of time. And I will end there, and I'm happy to take any questions.
Thank you so much.
Yeah, thanks, Tanya. What a tour de force.
You're welcome. Yeah, I'm actually looking forward to a time when I'm presenting some of the things from data we're collecting here at the NIH, but that's going to be a little while.
Yeah, yeah.
So I have a protocol in, yeah.
Let me see what we have here in the.
In fact, I have one question myself while Vince is checking for the questions.
Yeah, I
Sure.
see Armin has a question in the chat too. So you.
So how much we can associate the finding about the psychosis in schizophrenia in Dutch women to the malnutrition, how much could be potentially related to a stressful event situation that they are experiencing?
That's a really great question, because anytime, I mean, you have both that are occurring at the same time. I'm not sure I can differentiate the two. I mean, if you think of famine, that's a hugely stressful event, especially if you're pregnant. And so I would, I think it's hard to separate those. I guess if you had the same amount of or similar stress, but not the malnutrition, and had higher rates of schizophrenia. I'm thinking of, I mean, if you look around the world, there, at this time, there's many places where I think I would certainly not want to live there. There's high rates of stress where there might be enough food, although sometimes those two things, yeah, go together.
Yep. Thank you.
We have a question from Brad, which is, has there been any discussion of looking at climate or known pollutant data in Rotterdam during the prenatal period? And if there were significant changes between the collection periods, it would be interesting to compare these.
So climate, so there
Climate
has
or
been,
known pollutant, yeah.
so there have been a number of studies looking at pollution levels, and we have found, so we have found relationships with differences in brain, structural brain development, with decreased volumetric regions, and I think it's temporal, but I'd have to look up the exact areas. It's also a surface-based approach based on pollution. So we do have some of those. The climate change is an interesting one, especially for the Netherlands, because a third of the country is under sea level, and the Dutch are excellent at building structures to keep the water out, but there's certainly a limit. So I mean, the place where I lived, where actually I still have my apartment there, they said, I don't know, in 30, 40 years, it'll be, or at least I was on the 4th floor. So you know, the bottom level is going to be under water unless things are done. So there's an element of stress with that. But we haven't done it, as far as I know, we haven't done anything related to the, you know, the stress associated with climate change. It's been discussed as something that could contribute to mental health of populations. We did have questionnaires related to COVID.
There's a follow-up from Tricia, which is relatedly, there are neighborhood atlas data in these other countries similar to ADI or Childhood Opportunity Index in the US. How did the groups differ among gender or the presence of the Y chromosome? AZF?
What is AZF?
I don't know. Trisha? You want to unmute? I think you should be able to. Oh, maybe it's a spermia factor.
Okay. So we're
You're right.
really,
This is a different topic, but yeah.
yes. So first of all, let me just say, for Generation R, I use the language pregnant women, because I don't know of any in our group of. I'm very sensitive of in terms of gender diversity and all of that. But we didn't have any non-binary or trans men in the group. So just as a caveat, so I don't want to be insulting to anyone that I used, pregnant women. But we are very interested in sex effects, especially as you think about autism spectrum disorder that has rates 4 to 1 in males over females. And we've been, so a couple things. One thing that, as I have a grant together with Akgar Gasabian, who's at NYU, working with Generation R, we're looking at exposures to phthalates and bisphenols, but phthalates is very interesting because they're endocrine modulating. They modulate the endocrine milieu, and mothers who use certain products that have makeup and things like those type things, products, they can have higher exposures to phthalates, which may affect the endocrine testosterone and estradiol, estrogen levels in the development. And so actually we're working together with Vince and Akgar and Aloy Grignard to develop an index, actually to replicate some of the things you've done in ABCD into Generation R, to try and have an index of brain sex that we can then use to measure is there an influence of this phthalates on the structure of the brain. And many studies that we've done, we'll look at sex differences either by looking at interaction effects, or in some cases stratifying to see if there's differences. But autism is one thing that's very interesting from that standpoint. Did I answer the question? There was the other one. So we don't have. I really wanted to know what the rates were of children with intersex. We have looked at studies of gender diversity, those within the population, and then the relationship between gender diversity and autism, which we found in Generation R. And interestingly, for that, when we control for behaviors such as the child behavior checklist, we still see the effect. So even controlling for the total problem score of the child behavior checklist, there's still a relationship between gender diversity in children with autism spectrum disorder, and we're following that up now. And we've looked at brain imaging in that cohort as well. We don't have markers of like active SRY gene present or not, as far as I know.
Thanks. So I think we probably should stop for the day, but Tanya, for this Brain Space initiative, we do have the talk recorded, and we'll have a follow-up channel, Slack channel for discussion questions. We can put them in there, and use that as an opportunity to keep this conversation going. And the idea is hopefully that might lead to some maybe new ideas, collaborations, whatever. So we don't have to stop here. We're going to stop here, but we'll keep this going. So
Okay. Well,
yeah.
I just want to say thank you very much for the opportunity. If anyone has any specific questions, feel free to email me, and I'll try and get the Slack account, and I'll get on to the Slack account. And I'm curious what people think of this whole stochastic thing. I mean, are you more in the Einstein camp, which is deterministic, or are you more in the Niels Bohr camp,